Good evening, everyone, and greetings from Fatima Mother National College, Kollam, Kerala, India. We extend a very warm welcome to all those who have joined us here today. As part of the National Science Day celebrations, we had conducted a couple of competitions, a quiz competition and a caption contest, which were conducted over the last 2 days. And today we are culminating with the international webinar on Khorana and the genetic code by Professor Umesh Varshney. My name is Dr. Sushina Nathan, and I'm an assistant professor in the Department of Zoology at Fatima. Let's begin the program with a silent prayer. On that auspicious note, I now cordially invite Mrs. Nisha Thomas, the head of the Department of Zoology and convener of this program, to welcome the gathering.
Good evening and welcome all to yet another National Science Day celebration being organized by the PG and Research Department of Zoology in association with IQAC. So as we are all aware, we are currently suffering the vagaries of our own actions. And we have now come to a realization that sustainable approaches are indeed the need of the hour. So in this regard, this year's theme, integrative approach in science and technology for a sustainable future, is very relevant. So we have with us a very eminent personality to speak on, as Dr. Shoshina mentioned, Khorana and the genetic code. But before we go on to that, let me warmly welcome all our dignitaries who have joined the session. First and foremost, I would like to welcome our principal, Professor Jojo PJ, who has always been a strong support in all our activities and endeavors that the department has undertaken. And he has always given us the confidence to embark upon any activity. And it is in this regard that we have been able to organize this program today. And on behalf of the department and in my capacity as the IQAC secretary, I very warmly welcome our beloved principal, Dr. Jojo, to this session.
Thank you, Misha.
Thank you, sir. We also have with us Father Abhilash, who will be joining us. He has always been a strong source of inspiration and has always found time to attend almost all the programs that have been organized in the college. He has always made his presence felt with his very humble words of wisdom and has always a strong sense of support. I would call him our beacon. Welcome, dear Father, to our session. We have with us a very eminent personality, Professor Umesh Varshney, a renowned researcher, and he will be talking to us on the topic that is Khorana and the genetic code. Dr. Shoshina will be introducing him to us all. And so I would restrict myself by just saying that I sincerely hope, sir, that your deliberations will steer our children towards taking up research very seriously. And on behalf of the Department of Zoology and on behalf of the IQAC, I very warmly welcome you for having found the time to join us and to speak to our children. Thank you very much, sir, and welcome to this session. As with every session, our beneficiaries are our dear students and faculty of not just our college, from colleges around Kollam, and I understand from also Bangalore, various colleges of Bangalore. Welcome all for joining this session, and I hope you will benefit from this session. Thank you, and once again, a warm welcome to all of you.
Thank you, ma'am. I now cordially invite Reverend Father Dr. Abhilash Gregory, the Pro-Manager of FMNC, to deliver the benedictory address.
Good evening to all. It is my pleasure and privilege to appreciate the Postgraduate and Research Department of Zoology of Fatima Mother National College and IQAC for conducting a webinar to commemorate the National Science Day 2022 in a worthy manner. The topic of the seminar, Khorana and the Genetic Code, will arouse interest and enthusiasm in any science student. Hargobind Khorana, the Indian American biochemist. Hello, am I audible?
Yes, Father, you are audible, Father.
Yes, Father.
So his contribution provided essential information and technology for genetics and molecular biology, helping to lay the foundation of the incredible development of gene therapy. Today, we are fortunate to have Professor Umesh Varshney, Dean of the Faculty of Science, Indian Institute of Science, Bangalore, to speak about him and genetic code. Dear Professor Umesh, I am more than happy to welcome you once again and express my great gratitude for finding time to share your wisdom with us. I'm quite sure that your talk will certainly encourage our students to take science seriously and celebrate science. Without science and the developments it offers, we cannot move forward comfortably. We need more young people with more inquisitive mind, research aptitude, and scientific approach. May this webinar facilitate the listeners for the same. May God bless you all. Thank you.
Thank you, Father, for those inspiring words. I now respectfully invite Professor Jojo Pite, the Principal of FMNC, to deliver the inaugural address.
Thank you, Dr. Sushina. Honorable guest speaker, Professor Umesh Varshney, Dean, Faculty of Science, Indian Institute of Bangalore, Reverend Dr. Abhilash Girigiri, Co-Manager of the College, Mrs. Nisha Thomas, Head of the Department of Zoology, IQAC Coordinator and the team members, Dr. Sushina Nathan, Program Coordinator, Vice Principals, Deans, Heads of Departments, Faculty members, researchers, students, and well-wishers. A very good evening to all of you. The theme of National Science Day 2022 is Integrated Approach in Science and Technology for a Sustainable Future. Even though the International Science Day is being celebrated on 10th November every year, we, our country, celebrate Science Day on 28th February, commemorating the outstanding discovery of Raman effect by Professor Chandrasekhar Venkataraman in 1929. To mark this day, researchers and students in various institutions organize public speeches of scientific interest, radio, TV shows, exhibit scientific movies, science exhibitions based on the themes and concepts, and the like. This celebration honors Sir C.V. Raman for his exceptional contribution to research and the discovery by providing hope and inspiration to future generations to pursue their dreams. Various departments of our college are organizing seminars, webinars, and other activities to celebrate National Science Day in a befitting manner. In fact, the celebration of the Department of Zoology of our college is already on from 25th of February with a caption contest on the theme of sustainability and an online science quiz entitled Sustainophilia held yesterday. Today, we have the third event, an international webinar entitled Khorana and the Genetic Code. I would like to congratulate the Department of Zoology and the IQAC to find a very apt theme. And moreover, we are blessed to have a very eminent and well-known professor from the most sought-after institution in the country, the Indian Institute of Science. I sincerely extend our gratitude to Professor Umesh Vashni for being gracious enough to find time to talk to our researchers and the student community. Genetic code is a way our DNA is interpreted to create proteins. Dr. Hargobind Khorana opened the secret language of DNA. Decoding our DNA helped us to link sequence functions in our proteins, which illustrates the factors that controls our health. His discovery helps us to decipher the genetic codes. I'm sure Professor Washington will take us to the ocean of genetic coding and on the intricacies of genetic codes. With these few words, with the permission and blessings of all who are attending this webinar, I declare the webinar open.
Thank you.
Thank you. Thank you very much.
So I'm very happy and excited and honored to introduce to you my postdoc mentor, Professor Umesh Vashni. I worked as a Kothari postdoc in his lab for 2.5 years, and I have to say that those were the most intellectually satisfying years of my research career. And I think that is a testimony to the great scientist and teacher that he is. So after completing his BSc and MSc degrees in India, Professor Vashni went on to acquire a PhD in biochemistry from the University of Calgary in Canada in 1985. Thereafter, he completed his postdoctoral studies at the Massachusetts Institute of Technology, MIT, in the US. And then Professor Vashni joined IISc as an assistant professor in 1991. He was promoted to associate professor in 1997 and then to professor in 2002. He is presently a J.N. Tata Chair Professor in the Microbiology and Cell Biology Department and Dean of Faculty of Science at IISc Bangalore. Professor Vashni is doing outstanding research in unraveling the unique mechanisms of protein synthesis and DNA repair in the model organisms E. coli and Mycobacterium tuberculosis. He has over 220 publications in peer-reviewed and high-impact factor journals and has guided 28 PhDs and still counting. Professor Vashni is a recipient of numerous awards, recognitions, and honors. We will not be able to complete this webinar if I begin to list all of them now, so I will just mention a few of them. So was awarded the first National Bioscience Career Development Award by the Department of Biotechnology in 1999. He received the Shanti Swarup Nobel Prize for Science and Technology in 2001, which is one of the highest science awards in India. The Life Sciences Research Award of Novo Nordisk Education Foundation in 2002, the J.C. Bose National Fellowship by the Department of Science and Technology in 2008, the G.N. Ramachandran Gold Medal for Excellence in Biological Sciences and Technology in 2014, and the Goyal Prize in 2018 for Outstanding Research in the Field of Science and Technology. He is a fellow member of many societies and professional bodies and academies, including being a fellow of the World Academy of Sciences, TWAS, Italy. He is a chair or member of task forces of many funding agencies, research advisory committees, governing councils, and sectional committees. He is also an editorial board member and reviewer of many international and national journals. And last but not least, he is the convener of the joint CSIR UGC NET exams in life sciences. Needless to say, Professor Vashni has delivered keynote addresses and participated in numerous conferences across the globe. So we're indeed very privileged and honored to have Professor Vashni deliver a talk for us today. On behalf of Fatima Fraternity, I proudly invite our distinguished speaker, Professor Umesh Vashni from IISc, to present his talk on Khorana and the genetic code. Over to you, sir.
Thank you, Soshina, and thank you all my colleagues who have spoken before Soshina. It's a pleasure for me to be here. And I should say that it's only my students who can make me work hard. And right now, of course, we are in the midst of our lab symposium. And of course, I'm doing this talk from a place called Chalakere. And so I hope the connectivity remains fine. I hope I'm heard all right as of now.
Yes, sir.
Okay. So we will, with that, I think, let me just put my presentation on. Very good. Good evening to everyone. And what I thought I will do is actually give you a sort of a historical, account of how Khorana, from very humble beginnings, went on to achieve highest standards and highest achievements in science that allowed him to be respected worldwide. And in fact, he himself created a situation that people use him as an example to many, many things. And so the title of the talk, of course, is Khorana and the Genetic Code. And I'll probably be talking more about Khorana and less about genetic code, although there is a lot to be talked about genetic code as well. So this also is the centenary of his birth year. And so I would really like to dedicate this talk in favor of the celebration of the birth centenary for Govind Khorana. And this is a picture that was taken in a party at his Boston home, where he was just relaxing. And of course, I was able to take this picture there. Now, many of the things that I will talk about in this presentation, I have actually learned directly from Khorana or from a long associate of his, Uttam Raj Bhandari, who was my postdoc mentor. Of course, the two groups, the Khorana's group and the Raj Bhandari group, were sort of joint groups in the sense that we have joint research presentation, and so whatever I do, Khorana would be able to give me the feedback, and whatever people who work directly with Khorana, Raj Bhandari will be able to give feedback. But Raj Bhandari is a person who has been associated with Khorana from 1960 onwards until Khorana passed away in 2011. So this is a picture of Raj Bhandari and myself. So you're really seeing a sort of transition here. And I've also put up a picture that you're all familiar with. That is a Google doodle, which Google celebrated, Khorana's birthday in 2018. So this is really a very strong recognition of what Khorana was. Now, let me begin with a quote. And this quote, many of you would have heard. And this is a quote that was originally, I have learned it from a book that was prepared by Jawaharlal Nehru or written by Jawaharlal Nehru. Obviously, the highest type of efficiency is that which can utilize existing material to the best advantage. And I have slightly modified this quote by adding a couple of these words here. And this now reads, obviously, the highest type of efficiency is that which can utilize existing material and available opportunity to the best advantage. And I should say that Khorana is a very good example of this quote. And later on, I will show you the quotes that Khorana has created, which are also something to really pay attention to. And if one follows those, success is a sure thing. Now, this slide actually shows Khorana's career map or the journey of Khorana. Khorana was born in a small village, Raipur, which is now in Pakistan. So before there was division, Raipur was part of India. Of course, now it is in Pakistan. He was born on 9th January 1922. This is the date that is a probable date of his birth. People don't know the exact date of his birth. His father was a Patwari, which I'm sure many of you will recognize. They really don't earn a lot of money, but the important thing was that his father wanted, he himself was interested in education and wanted his children to be educated. This village of Raipur is a very small village. At the time, there were about 100 people in this village. That's all that was there. And luckily, it had a school where Khorana went to study. Now, this school also did not have any buildings. So Khorana and other colleagues of his studied by sitting under any shade, under tree or any other kind of shade, and learned from the people who were teaching, whatever they were teaching, based on that, learn from there. And so this is really something which requires a lot of dedication, where it is not a very systematic education. There is education, but then I think it all depends on the hard work and the commitment, the dedication that the student can show. And this Khorana did show. Now, when you don't have a systematic education, there are a lot of deficiencies that one inherits. And Khorana was aware of those deficiencies, and he always wanted to make up for those deficiencies. He did well in his 12th standard, and that allowed him to go to Punjab University in Lahore. He did his bachelor's, and then, of course, 1943 to 1945, he did his master's degree. After he had done his master's degree, I'll come back to many of these details in a little bit more descriptive form. But what I would now like to show you is that when he had completed master's, remember this is still pre-independence time, 1945, he did well, and the government of India at the time awarded him a fellowship to go and study in UK. The fellowship was awarded to him to study insecticides and pesticides, or fungicides actually, insecticides and fungicides, because the fellowship actually came from Agricultural Ministry of Agriculture. When he went to UK, this is also the time when a lot of the war veterans were returning and going back to school. And so Khorana could not be placed to study insecticides and fungicides. The High Commissioner in UK then decided that because Khorana has masters in organic chemistry, he should do fine in any organic chemistry course. So this is where he got admission in Liverpool, and he studied organic chemistry. So this is one example where a bureaucratic decision turned to be something which was right decision. Often the bureaucratic decisions don't turn out to be right, but this was one where it yielded really the desirable results. After he completed his PhD in 1948, 1949 area, actually 1948, he completed his PhD. And at that time, or in those days, much of the literature or the scientific literature that came out, particularly in chemistry, used to be in German language. And so when he would go to the library, many of the research publications would be in Germany. So he decided that he should actually go to a country where he can learn German so that he can be a lot more proficient in Germany, and as well as he can follow the literature quite well. Now, this is really quite interesting, and I will come back to this one again, But from Liverpool, he went to ETH, where he did 1 year postdoc, and he did only 1 year postdoc because he was not getting any fellowship. He did the postdoc simply based on the savings that he had from his PhD degree. So in 49, after he returned to India. So you see his first journey is to Liverpool, second journey is from Liverpool to Zurich, and the third journey is from Zurich back to India. Now, 1949, this is after the India had divided into Pakistan and India. And you can see that this is immediately after independence, 1949. There were really no jobs. He spent some time in India, and a lot of the people, his friends and relatives, in fact, were in Delhi area, simply because they didn't have job, and sometimes some of them actually were living as refugees. So 1949, he spent some time in India looking for jobs, but he did not find jobs. But luckily, he had actually in Zurich established a relationship, a friend who actually then said that, look, you could come to Cambridge. There is a postdoc position available, and I'll come to all of these details again. So then he went back to UK. He did, postdoctoral work there. And in UK, he actually met Cambridge. He met a person who from University of British Columbia, who was looking for people to hire to a completely new setup in UBC. Khorana at that time did not know where Vancouver is, but he accepted the job, looked the word map, found out where Vancouver is, and went to Vancouver. After doing some work in Vancouver, he actually did very well there. He established himself, and then University of University of Wisconsin in Madison, they offered him a job. He stayed there. The work that he did there earned him the Nobel Prize, and after that, MIT in Cambridge was very interested in getting him to Cambridge in USA, and he moved to USA, and that's where he remained. So I now want to just, giving you this summary. I will come back to many of these details, but I want to now show you his own writing, where you will realize how modest he was. He writes about himself in an article that was published in Science in 2000, and that says, I have met people who believed they knew at a very young age what they wanted to do in their lives. I envied them, but my own life was not like that. And this is a quote that he has given, and I'm actually extremely influenced by this quote because it actually opens up or provides a way for people. Many times people think that if they don't know what they're going to do 10 years down the road, 15 years down the road, it's actually okay. And this is what Khorana did. He did not know what the next thing is in his career. The bottom line really was that you work hard and make use of every opportunity that has been made available to them, and he did so well. So I think this is a very good statement that he has made that really opens up, encourages everyone to do science. So this is one thing that I've been extremely influenced by his statements. Then you will see that it really goes back to convince you about the quote that Jawaharlal Nehru has given, and which I have modified slightly to fit the context. He himself says that at age 18, when I sought admission to the Punjab University in Lahore, I applied to 2 departments, English literature and the honors course in chemistry. Admission into the latter, that is in the chemistry, was restricted and required an interview. My shyness kept me from in the interview, but they saved a place for me anyway, and thus I became chemist. So again, you see that his intention was to either join English literature or chemistry, as the chance will have it, he became a chemist because the chemistry department, in spite of him not appearing for interview, offered him an admission. So basically, he got the opportunity to learn chemistry and he became a very thorough chemist. After I received my PhD in 1948, as I said, in Liverpool, these are his words. I was eager to spend some time in a German-speaking part of Europe, and I managed to do this at ETH in Zurich, Switzerland. I worked with Vladimir Prelog. He was inspiring, and so was the Institute with its long tradition of excellence in organic chemistry. So after doing his PhD, he was really determined to study in a German-speaking country, and that was what brought him to ETH. Now, another story is even more interesting here. And the story here is really that he just walks up to the lab and says, here is my thesis, no recommendation letters, no nothing. In his own words, what you're seeing here is, I arrived in Zurich one day in the late summer of 1948. I left my luggage at the railway station, took my PhD thesis with me, and went looking for Professor Prelog. He had no idea then that an Indian student was about to approach him to plead for a little space in his laboratory. No one from Liverpool has sent out any letter of introduction or recommendation on my behalf. When I met Professor Prelog, I found him so accessible that I immediately lost all fear. I requested that he might look at my PhD thesis. This was an organic nitrogen compound and alkaloid on that topic. And I would come back the next day to ask if he would accept me in his lab. Next day, Professor Prelog, with his typical enthusiastic and encouraging smile, said yes. And at my excitement, added that he was glad to see people happy. Looking back now, over the years, I believe that spending a year at ETH with Professor Prelog is probably the wisest thing I ever did in my life. In fact, after he left his lab, he has always made a point to visit the lab of Professor Prelog whenever he had the opportunity to do so. Now, during this time, he actually was, of course, spending time in the library. And he discovered a very, very old literature or the science in the Journal of Chemistry, a reagent, carbodiamides, so dicyclo, carbodiamide here. This dicyclohexyl carbodiamide, this he realized was a reagent. It's a mild reagent, but he could make use of it to make nucleotides or actually make many other compounds using this reagent. So I spent a great deal of time studying. It was then that I came across this particular reagent here. Serendipitously, carbodiamides became important in my synthetic work later on. So this was something that he discovered. It was actually, work was done long, long ago by making use of this reagent. Nobody paid attention to it, partly perhaps because it was written in German, but he was able to realize the importance of a reagent like this. This is, I won't read the whole thing, but it really sets a stage by when he says that, 1949. So he joined a Prelog lab in ETH in 1948. And as I mentioned to you, he was not getting any fellowship. And again, really shows the kind of determination he had to study chemistry. Whatever savings he had, based on those savings, he worked in the lab. And in fact, I think everybody was highly impressed with the manner in which he worked in that particular lab. In fact, within a matter of about 11 months, he published 2 papers from there. And those were actually written in German. So 1949, as I mentioned to you, after doing that postdoc, he returned to India. And I already told you the story that in India, there were no jobs. So, he returned to, Cambridge, UK. And so he was working there in the Todd's lab, Alexander Todd's lab. And at that point, he met a person, Gordon Shrum, from British Columbia Research Council, while doing postdoctoral research in Cambridge. Dr. Shrum told Khorana that he had very little by the way of facilities, but there was all the freedom to do whatever he wanted to do. Khorana did not know at the time where Vancouver was. He accepted the job, and when looked at the world map to find out where Vancouver was, he moved to Vancouver, established an outstanding research program. His research on nucleotide synthesis brought famous scientists like Arthur Kornberg and Paul Berg to his lab as visitors. So this was the kind of work that he was doing. Now, Arthur Kornberg, most of you will realize, worked with DNA polymerase, got a Nobel Prize. But for him to do his work, he needed nucleotides, deoxynucleotides. And who would make them? Who has the technology to make them? Khorana, of course. So Arthur Kornberg visited his lab in Vancouver. Likewise, Paul Berg, he needed to make amino acyl AMP because he was at that time working with tRNAs. And so again, how would he make amino acyl AMP? Khorana's technology. So he came to Khorana's lab, and both of these people became very good friends and had a lot of influence action that Khorana then subsequently took to make use of the chemistry to study things that relate to life sciences. Now, here you have Arthur Kornberg. Now, this is picture taken in a party that was held at Vancouver much later. But I put this slide over here just to show these 2 great scientists, Arthur Kornberg and Paul Berg. And of course, in the middle, you have Khorana. So they are really having some good time. This is a conference that was held in honor of Khorana. I believe it was in 1993. And all of these people are together. So this friendship that he developed actually allowed him to move into biological systems. And of course, Khorana was also visiting the labs of both of these. And he subsequently made use of DNA polymerase for his own research. Now, at Vancouver, this is Khorana's group at Vancouver in 1959. And I put this picture particularly just to show you this one person. This is actually Mike Smith, who actually got a Nobel Prize for the work that he did on site-directed mutagenesis. So Mike Smith was a postdoc of Khorana. So there is sort of a legacy, and I'll show you that many of the people that Khorana worked with also got the Nobel Prize. His postdoc also got the Nobel Prize. He himself had the Nobel Prize. And these are the pictures of Vladimir Prolog that I have been talking to you about. He got Nobel Prize in 1975, of course, after Khorana. And Alexander Todd, that he worked in University of Cambridge with, he got the Nobel Prize in 1957. So you see the kind of lineage that he comes from, and the lineage that follows from the group of Khorana is very, very eminent lineage. So much so that people actually have created, monuments kind of things, for Khorana. So this is a Khorana Park that is there in Vancouver. He has received so much respect for what he has done in science, the way he has influenced science, and the way he has trained the people who have worked with him. He's earned the kind of respect, and that respect is really something that really is highly, highly memorable situation. And it's actually, Khorana is a legendary scientist, and you can see that in his honor, city of Vancouver has a park which is called as Khorana Park. There are many other things that have happened. So in Vancouver, he was there until 1960. In 1960, as I mentioned to you, he moved to University of Wisconsin, where actually they created a sort of institute for him, Institute for Enzyme Research. And that is where he did a lot of the work that led to award of the Nobel Prize to him. That quite a bit of work was actually done at University of Wisconsin. 1968, as you all know, he got Nobel Prize in medicine or physiology. He carried out chemical synthesis of the gene and established his biological activity, the first example of synthetic biology. So most of us know Khorana for the genetic code, but in fact, there are areas that are fashionable areas now. But if you go to the history of these areas as to how and who established them, you will find that it was Khorana. To show that a chemically synthesized gene works in the cell is a work of synthetic biology, and he is the one who established that. There are a lot of the fields that he has started. The chemical biology is a field that originated just because Khorana worked on the topics that he did. So this is really something very outstanding that Khorana was able to do. Now, I will say a little bit about the work. I don't want to go into the technical details because I'm told by Soshina that there are people from high school also. So basically, I will not go into the technical details, but still tell you something about the work that led to the award of the Nobel Prize. So he was able to develop a technology, a methodology to make short polydeoxyribonucleotides. And those he was then able to, as I said, that he was able to make use of DNA polymerase, which he learned using from Arthur Kornberg. He was able to make use of DNA polymerase to make long polydeoxyribonucleotides of known sequences. Those sequences, he was then able to make use of RNA polymerase to convert into long RNA. And that RNA, he was then able to use it as mRNA or the template for making protein. And of course, the technology to sequence protein was available, and he was able to then find out what the sequence of the polypeptides that he has made is. So this was the outline of what he did. I'll just give you a little example of, so that you can get a feel for it. So he had developed the technology of making short oligonucleotides. So here it is a dinucleotide, but it is a polymer of dinucleotide, UC. And when he uses that kind of message of the RNA for in vitro translation, he got polypeptides which had alternating serine, leucine, serine, leucine, serine, leucine. Now, if you pay attention to this one, I guess this is being covered a little bit here, but okay, I can't get rid of it. So you can have two possibilities here. You can have UCU, CUC, UCU, CUC. You can have only these two alternating codons. So now you already know that one of these codons codes for serine, another one for leucine. You don't know which one of them codes for leucine, which one of them codes for serine, but this gave the idea that one of the codons is for serine, another codon is for leucine. He can then make another dinucleotide polymer, UG, and that when he translates gets valine and cysteine. So again, he can have an idea that these two codons code for valine or cysteine. Likewise, ACA, CAC can code for threonine and histidine. These three codons, isoleucine, serine, and histidine, GU, AU, AG, AGU, serine, and valine. So he was able to tell that this particular group of codons will code for this particular group of amino acid. Now, clearly, it tells a lot about the codons as to what they code for, but it still does not identify a given codon coding for what amino acid. So this actually was developed further, and this is where some of the work that Marshall Nirenberg did, who was also, who shared the Nobel Prize with Khorana, came in use. So now let us say we have a situation where there is a triplet sequence that is available. Now this triplet sequence will bind to a tRNA which will be complementary to this codon. And if the tRNA is amino acylated, one would know what amino acid has bound to the ribosome. And in doing so, he was able to now define if he is taking AGA, he's taking GAA, he's taking AAG. Now you can see that when he takes AAG, lysine binds. When he takes AAA, then also lysine binds. So now he is able to tell that AAG and AAA, they both code for lysine. So by doing this kind of exercise, this is actually huge exercise when you try to do by making use of all the 64 triplets. After doing this kind of work, he was able to establish this genetic code table that all of you are familiar with. Now, of course, it is told that these genetic codes or the codes are fixed, but that is not so. The genetic code is evolving code. Many of these codes have different meanings in different organelles or different organisms. And a revised genetic code table, which again, I will avoid going into the details, but if one thinks that the genetic code is completely solved, it is not. It is a field that is still evolving, and there's a lot more to do in this particular field. I will now briefly tell you about this polymerase chain reaction, which we know. In fact, I think Khorana is the one who discovered polymerase chain reaction. Of course, he did not get credit for it. He should have got a second Nobel Prize for discovering PCR. And I'll tell you why he should have got the this second Nobel Prize. What he had done was he had actually made short oligomers. He made complementary, he denatured those, and by making use of primers that will anneal to one end of this oligomer, he was able to amplify these oligomers by multiple cycles. The only difference here is that he used DNA polymerase, which was not a thermoresistant or polymerase. So at each round of cycle, he had to add fresh aliquot of DNA polymerase so that he could amplify. This is really what I'm showing you here is nothing but PCR. He did not get credit for it, but he is the one who actually discovered a polymerase chain reaction. Now, if we move further, by developing this technology of gene synthesis further, or polymerizing the nucleotides into a polymer, he actually started synthesizing a gene. This is the time when the definition of gene was not at all known, 1970. He published this work. But what was known was there was a sequence of alanine tRNA, which was done by Hawley. And in fact, Hawley also got the Nobel Prize along with Khorana. So Hawley, Khorana, and Nirenberg, three of them got, shared the Nobel Prize. Hawley was the one who actually was able to determine the sequence of tRNA alanine. And at that time, the promoter, nothing was known, but Khorana started synthesizing the gene and left a space for putting in promoter. But he did the complete chemical synthesis of a gene. Now, this paper is published in Nature in 1970. This is actually a summary paper. The details of this work, the full details were published in Journal of Molecular Biology. And Journal of Molecular Biology at that time was really the top journal in the field. He had 13 back-to-back papers. The entire issue of JMB was dedicated to the work from Khorana's lab to describe the synthesis of this alanine tRNA. So this is Khorana actually doing this kind of work here. Khorana always worked on the problems that were at the interface of chemistry and biology, but chose them carefully, and the methods to tackle them were always well thought out and futuristic. You can see that the kind of technologies he developed while he did his own work were all very, very futuristic technologies. And in doing so, he defined a new area of research, which we know by the name chemical biology. Here is Khorana receiving his Nobel Prize from the King of Sweden. And these are the three people that I've been talking to you about, Hawley, Khorana, and Nirenberg, who shared the Nobel Prize. Other than Nobel Prize, of course, Khorana had a lot of recognitions. I won't even go through the details of it, but we'll notice that the government of India also awarded him a Padma Vibhushan after he got the Nobel Prize. And in fact, the Indian Academy of Sciences, Bangalore, gave him the fellowship of the Academy. There are many more awards, which, once you have got the Nobel Prize, you don't need to talk about the other awards. So after he got the Nobel Prize in 1968, MIT made an offer to him to offer a Alfred P. Sloan professorship, which was a joint appointment between biology and chemistry departments. He joined there in 1970 and continued at MIT until his death in 2011, November 2011. Now, what Khorana also did, in alanine tRNA, he did the complete gene synthesis. But to show that chemically synthesized gene works in the cell was done by developing a suppressor tRNA for tyrosine, where again he used the same technology of synthesizing short oligonucleotides and then joining them together. And again, he did not know at the time the promoter sequence, but he left a space for putting in promoter sequence. And after the promoter sequence was put in there, he was able to show that the chemically synthesized gene was functional in the cell, in bacteria. So this was really the first example where a chemically synthesized gene could be shown to function in biological system. Now, at this time, he's already very, very famous in this particular field. But at this point, he then decided to work on another very difficult problem, which was relating to membrane proteins. Now, membrane proteins are very, very difficult to purify. And not only purified, but then to show that they can be functional. So he actually started working with bacteriorhodopsin, and rhodopsin from mammalian systems, and he did a lot of work with this one. These two proteins established a completely new field. And again, of course, I won't go into the details of it, but suffice to say that at every juncture, he chose to work on difficult problems, but of course, extremely good record keeping. So if there was any failure in the experiment, he could always go back to the records and find out where the difficulties were. And if difficulties were not there, if there were no errors made, to then figure out a new way of achieving the objective. Khorana's group, every year there are pictures taken in the group. So there are a lot of social activities also that happened in Khorana's group. And this is a picture in 1990, just a year before I left MIT. And you can see here I am. I guess most of people have difficulty recognizing. That's me, Khorana, Raj Bhandari, many of the other people from both the groups are over here. Then again, I was visiting MIT at one point. This, I think, is around 97. I can't remember, but again, you can see the group is somewhat smaller, but again, it's a joint group, the joint activity. Not only these group meetings, but, Khorana had a sort of, house about 2 hours drive from Boston, a farmhouse kind of a thing. And he welcomed people. So, here you see a picture where he has reached before everybody has reached to the same place. And he's holding a placard there to show how to get to Khorana's house. So you see, how nice it is of him to reach there early and then making sure that people reach the house, without any difficulties. There are parties going on. This is my daughter here. Can't see her face. My wife, Archana, here. And this is, of course, Khorana, and is, talking to all the people. There is a food all over. People are eating food. And, there's another picture of the same thing. The party going on in his house. this also happened to be a way of, this apple, picking also was quite popular. So this is a picture where, one could go for apple picking. The group also had activities, of
Kindly unmute, sir. It's onto the mute. Sir, the audio is muted. Kindly unmute. Thank you.
I done. Okay, well, good. Okay, so where were we? Yeah, so this is the, when we have gone for deep sea fishing. So picture taken on a ship, and to show that, it's not just a photo op. I did catch some fish. He's a fish, one of the fish. I think I caught some 3 or 4 fishes that day. And this is one of them. So not only this, there used to be quite a few symposia that were held in honor of Khorana. And this is a symposia that was held in 2000 in Brandeis University in Waltham, very close to Boston. And he's with his people. And during this meeting, in fact, actually, he also presented gift to people. And this is a chemical biology book where he has actually put his own publications, own papers that developed the field of chemical biology with his own summary of those papers. And he sort of gifted the book to people who attended the meeting. And this is, with his own personal notes on the book, he was, able to give these. So he actually cared quite a lot about his people who worked with him and was always appreciative. This is another picture that was taken in Japan, another symposium in 2004 that was held in his honor. There was yet another symposium in 2009 that was held in his honor in University of Wisconsin-Madison. There was also, another symposium. Now, 2011 symposium that we did at ISC. This was actually held in honor of Raj Bhandari, but unfortunately, during this time, Khorana passed away in November. And when that happened, because most of the people who were at the meeting were associates of Khorana as well, we held a special session in memory of Khorana. So in fact, this symposium then became a joint symposium for both Khorana and Raj Bhandari. And the proceedings of that symposium were brought out in an issue of Resonance published by Indian Academy of Sciences, Bangalore. And again, of course, you can see a cartoon where these 3 laureates have been now sort of, cartoonistic representation of codon, anticodon recognition. There was yet another symposium that was held in Chandigarh in, I think this was 2017, I think. Is the date written? Yeah, 2017. And so this was also a very good symposium. Now, I want to sort of, end it soon. What I want to show now is that his recognition and respect for him is so much so that many of the organizations have started many things, which are after Khorana. So there are many professorships that have been, named after Khorana. This is a scholarship program that Government of India, along with an organization in US, WinStaff Forward, and Indo-US Science and Technology Forum. They all together offer this fellowship, and the students are selected from within India. Their travel is paid for by Government of India, but then they are placed in different laboratories in United States, and their stipend there is covered by the labs that they work in, and they get good training. Often these students will go back and work PhD students. So this is a very nice program that has begun. And Khorana had the opportunity of actually interacting with many of the students who went through this scholarship program. Now, I want to, before I end, I want to show you some of the quotes that Khorana from time to time has been giving to people. And now, while I've heard many of them, but many of them actually I have adopted from a talk that was given by my mentor, UL Raj Bhandari. And if you look at it, be determined and self-motivated. You shouldn't be waiting what your mentor has to tell you. Mentors are there, but you have to be determined and self-motivated to do what you want to do. Be bold and aim high. Be disciplined and work hard. Always do your best. Strive for excellence. Stay focused. Do not get distracted. And if you remain focused, you will solve the problem. Plan carefully. Prepare yourself well. Be thorough. Follow every lead. And be thorough means you also write down everything very clearly. And when you write down everything clearly in the notes, you'll be able to follow the leads, all the leads that you need to follow. No shortcuts. Do everything necessary to prove a point. Perseverance is another something very important. Do not give up. And finally, if you want to get far, you have to travel alone. That is what Khorana always said, that if you have to do something novel, there will be a lot of people who will say that it's not going to work. And this is really where you need to travel alone. You have to travel far. That's really some of the things are, the course are real practical course. He has lived up to these courses on his own, and he's giving this course or giving these, advices to people who have worked with him based on what he has done. It's not something that he's telling things, as he has heard. So with those words, I'd like to thank you all for the opportunity given to me to talk about something I really love to talk about, that is Khorana, and what I have learned from Khorana, and what many of us have learned and practiced what Khorana taught us. With those words, I'd like to end. Thank you very much.
Thank you very much, sir, for that very engaging and illuminating talk on the life and work of Hargobind Khorana. Truly from humble beginnings to greatness, he is a legendary scientist, and his life, his work is truly awe-inspiring. I'm sure this talk has inspired the students and the faculty alike who are listening to this to persevere and never give up and to pursue their dreams. We will now go ahead and take questions from the audience. Feel free to unmute yourself, or you can type your questions in the Q&A box.
And students, if you want to ask questions, kindly raise your hand so that we do not have more than one student asking the question. Thank you.
Please go ahead.
So, sir, I have a question actually. It's a technical question. Yeah, I was just wondering, how is an in vitro system different from the in vivo translation system? Because there you are able to translate the protein without the initiate the codon or initiated the RNA. I was just wondering, what is the, because like he started with UC, and he was able to get that translated. But in a,
I think you're asking me, how come the translation happened without a start codon? Is that the question?
yes, that's the question.
Right, that's a very good question. And sometimes not knowing the details is a big boon. At that time, they did not know. Of course, later on, he discovered the start codon and termination codons, but when they began, they did not know. And the reason it worked in translation was because this was done in vitro, and the conditions of translation was such that the selectivity for initiator tRNA was lost. So it was not a very high fidelity translation system. It happened without the presence of initiator tRNA. And that is something, as I said, was something not knowing was a help, because if they knew at that time, they would not even attempt to do it this way, the way they did.
Yes, sir. In fact, I don't think they would have discovered anything at all. I mean, if they had started off with
I think many times one shouldn't
serendipity.
worry about the things that one doesn't know. If there's a good hypothesis, if there's a good basis to do something, one should just jump in and do it and then figure out what more needs to be done.
Yes.
Soshina ma'am, there's a question in the chat box. It's from Dr. Jayashree Kurup. Sir, I'd like to participate in the teacher summer internship. I applied in IISc Bangalore, but never got selected.
That is the program that Indian Academy of Sciences carries out, isn't it?
Yes, sir.
But for last 2 years, I think there aren't very many people that have been selected because of this COVID issue. So that is also a bit of a competitive, and I think one should keep trying. I know there are many people who have gone through my lab, both students and the teachers. So yes, I think what you're saying really is that we need to have more opportunities for people. Currently, I think while we have the programs, they're probably not enough for everyone. Did I say it correctly?
I'm not sure for the next part of the question, sir, but I think ma'am has mentioned PDF, but I think it would be PhD, which I'm not sure. I would also like to do PhD, but age bar is there. I'm not sure if that's exactly the question that ma'am wanted to ask.
So,
I think it's PDF, sir. Postdoctoral fellowship. I think that's what you meant.
there is no
Yes.
age limit for that, but when you have younger people competing, that becomes a competition, I think. But there is no age limit for finding a postdoc fellowship. If you have a good idea, if you have a good project, and you have the background of doing it, I'm sure people would love to have you in the lab.
Thank you, sir. I think one of the student called Sandhya Shrikant wants to ask a question. Sandhya, kindly unmute yourself.
Good evening, sir. This is Sandhya here. I am a third year BSc student. I am studying biotechnology as one of my majors, and I'm very much interested to pursue in the field of research. So I wanted to ask if your lab offers any opportunities for internships that I can look out for.
Yeah, so my lab does offer, but many times, the people who come to my lab, they come through Indian Academy of Sciences or KVPY, or, there is a program from JNC ASR, where I'm honorary professor. I take people from there. So I do have commitments to many of these institutions. So I would strongly encourage you to apply to these institutions, and you're welcome to give my name as one of your options. And I'm sure, I don't discriminate people. So, if you have been selected there, you will definitely get an opportunity to work.
Sure, sir.
But
Thank you,
there are,
sir.
many other labs in the institute who do better than my lab. So you should actually also try in other places.
Sure, sir. I'll try, sir. And I also had a question about the explanation of the genetic code that you were talking about.
Yes.
So all the permutations and combinations are not there. So is that what you mean by everything is not found about the genetic code, and there's still
No.
possibilities?
That is not what I meant. In fact, there's a lot more work I have not talked about. If you're interested, actually, many of these details you can find in the articles that were written soon after his Nobel Prize. Nobel collection, you'll find articles where he has described many of these oligomers. But there was a limitation in doing this because you will realize that this amino acid can be coded for by more than one codon. And therefore, to assign size identity to those codons,
Renita, ആ ടാബിൽ ഒരുത്തന്നെ എടുത്തിട്ടുണ്ടോ നോക്കി.
therefore, the only way they could finally assign the identity was when they studied by synthesizing the triplet sequences and used them to capture amino acylated tRNA on the ribosome. So that then allowed them to identify all of the 64 codons as to, in fact, during this time only, it became clear that there are some codons which don't capture any tRNA. And so therefore, they then became a suspect for termination of the protein synthesis.
Okay, sir. Thank you, sir. Got it.
Thank you, sir. And we have another student, I suppose, Sonia Rani, who wants to ask a question. Sonia, kindly unmute yourself.
Yes, sir. Thank you, sir. Good evening, sir. This is Sonia from final year student from JNC, Jyoti University College Autonomous, Bangalore. So I'm doing biotechnology, but I want to do my master's in IISc. Unfortunately, due to some reason, I wasn't able to register for few competitive exams. So is there any other way through which we can get admission?
So in ISC, we are just beginning a brand new master's degree program. But I think you will have to come through JAM for this one. You know, there is a joint admission test program that you will need to appear for. I do not know whether the deadline for applying for that is gone. There is another program, which is integrated PhD program, where you can join after your undergraduate program, after BSc. But for that also, you need to have that JAM entrance to come through to ISC. There are no other ways. Earlier, we used to hold our own written tests. We have done away with them because there are now sufficient qualifying exams that many of the agencies carry out, and we choose the students, shortlist the students from those qualifying exams, and then we hold the interviews to finally select the people who come to our programs.
Thank you, sir.
Are there any other questions?
Not yet, sir.
Sir, I'll ask a question. Right. You were talking about the Khorana program for scholars. Is that only for chemical biology or?
No. This is for biological science, and many people can go from different areas there. In fact, that is now joined as a combined as a Bose and Khorana program. So you could, in fact, go for physics also.
I think we have a question from our principal. Sir, kindly unmute, sir.
Professor, good evening. I have no question, but I have to thank you for enlightening or igniting the minds of our students. The questions
My pleasure.
really